Assalamu alaikum, guys. We are going to start from where we ended up last time. We were discussing about UV visible spectrophotometry. Just to connect with what we were discussing with the today's lecture, the last topic that we discussed in UV visible spectrophotometry was about the electronic transitions, and I discussed with you that there are six possible electronic transitions which are listed in this slide. That is from sigma to sigma star, from pi to pi star, from non-bonding to sigma star, from non-bonding to pi star, from sigma to pi star, and from pi to sigma star. These are the six possible electronic transitions in UV visible spectrophotometry. I'm not going in the detail. It's just a summary that we discussed in the last time. So these were the examples we discussed in terms of how that transition takes place, what sort of energy is required for this transition, and with some examples of the compound. So we have already gone through these six types. So what we are going to start today is actually the terms used in UV visible spectroscopy. The first term that we're going to discuss here is about chromophore. I'm pretty sure that you guys are aware of the term chrome that refers to color. So, the part of a molecule which is basically responsible for color production or for the color of a compound is termed as chromophore. Or, in other words, any functional group which contains multiple bonds, and because of that nature of the multiple bonds is capable of absorbing the radiations which are above 200 nanometer, and they will do the transition from either non-bonding to pi star or from pi to pi star. Examples are given here, like nitrogen dioxide, then nitrogen and oxygen double bond, and carbon and oxygen double bond, and etc. etc. These are the compounds, these are the functional groups rather. They have multiple double bonds and they are capable of absorbing the radiations above 200 nanometer. So thereby they have specific colors and they can be referred to as chromophore. So, in order to understand or in order to interpret UV visible spectrum, there are following points that we must consider. The first point is the non-conjugated alkenes show an intense absorption below 200 nanometer. I'm sure that you know what are alkenes and what are non-conjugated alkenes as part of your organic chemistry. So the non-conjugated alkenes, they show an intense absorption which is below 200 nanometer, and therefore they can not be accessed with the UV spectrophotometer. So this one point has to be understood, that if the compound is capable of absorbing the radiations above 200 nanometer, typically in the range of 400 to 700 or 800 nanometer, then we can use visible spectrophotometry. While in case of the non-conjugated carbonyl compounds or carbonyl group compounds, they have a weak absorption between the range of 2 to 300 nanometer region. So you can see here the examples are given. One of the example is of acetone, which has the maximum absorption at 279 nanometer. And then we have another example of cyclohexane, where the maximum absorption is at 291 nanometer. So in this case, when a double bonds, they are in a conjugated position, the absorption uh, maximum is normally being shifted to a little bit of longer wavelength. So if you compare these above two examples with the other two examples of 1,5 hexadiene, that compound has a maximum absorption at 178 nanometer, and that of 2,4 hexadiene, they have absorption maximum at 227. So now you can differentiate that when we have conjugated double bonds, normally the absorption maximum is shifted towards the longer wavelengths. Now, what actually is this conjugation is doing? this conjugation of a carbon-carbon double bond or a carbonyl group actually is going to affect the absorption capacity of the compound, and thereby the absorption maximum will be shifted towards a bit of the longer wavelength. Two of the examples are given here. Ethylene, it has got a maximum absorption at 171, while in contrast to this, acetone has a absorption maximum at 279 nanometer. And we know that acetone has got the carbonyl group, as you can see here, in contrast to ethylene, which doesn't have. Then we have another example of crotonaldehyde. They do have a carbonyl group here, and you can see their absorption maximum is 290 nanometers. So now this point has been pretty much understood, that the presence of these carbonyl groups or a carbon-carbon double bond in certain cases as well will result in the shifting of the absorption maximum to a longer wavelength, as it is evident in this particular example. The next term that we're going to discuss is about oxochrome. Now, what are oxochrome? The functional groups which are attached with a particular chromophore. As a result of that, it modifies the ability of the chromophore to absorb light. So we know that chromophore itself, they have the capacity to absorb light. That's why we call them chromophore. But there are certain functional groups which, with attached with the chromophore, will affect its ability to absorb light or alter its, capability of absorbing light. That's what we call oxochrome. Or you can also define that as the functional group having the non-bonding electrons, which does not absorb radiation near the UV region, but when they attach to a chromophore, it alters the wavelength and intensity of the absorption of the chromophore itself. That's what we call as oxochrome. Here we have certain examples. Benzene has got a maximum absorption at 255. Now, with the incorporation of this OH group on the benzene, this is what we call the oxochrome. And as a result of this OH group, the wavelength maximum has been shifted to a little bit longer wavelength of 270 nanometer in contrast to benzene itself, which is 255. Another example of this oxochrome is this NH2 group. And as a result of this NH2 group substituted with the benzene, that is called as aniline. And you can see its maximum absorption has been changed to 280 nanometers. So that's what we call oxochrome. These are the functional group like hydroxyl group or NH2 group, which is attached itself with the chromophore. And as a result of that, it will alter the absorption maximum of that particular compound. Now, the next thing we're going to discuss is about absorption and intensity shift, because this is how the base of this technique is, because there is a particular absorption, and as a result of that, there is a certain change of energy level, and that results in the intensity shifts. So now we are going to discuss about that. There are four effects in this regard that we're going to discuss in absorption and intensity shift, and we'll discuss them one by one. The first one is called as bathochromic shift, which is also being referred to as redshift. The next one is the hypsochromic shift, which is called as the blue shift. The third one is called hyperchromic effect, and the fourth one is called hypochromic effect. Now, these are the shift actually. As a result of the absorption, the energy levels are changed, and because of the change of the energy level, there is a change in the intensity or a shift in the intensity, and that's what we call intensity shift, which just I have discussed with you. There are four types of that. So let me go in the detail of them one by one. The first one is a bathochromic shift, which is being also referred to as red shift. Now, what is bathochromic shift? When absorption maxima of a compound is going to shift to a longer wavelength, it is known as bathochromic shift or a red shift. Now, if you go back to the example I discussed a couple of minutes ago, the example of benzene, you have seen that benzene was having a lower absorption maxima, but as a result of the attachment of the oxochrome, there was an increase in the wavelength absorption. And that is what actually called the bathochromic shift. You can see here what we are saying, that when there is an increase or a longer wavelength, that is being referred to as bathochromic shift or the red shift. Now, this effect is due to the presence of an oxochrome, as I just mentioned you the example. Like benzene itself is a chromophore, and then there's an oxochrome attached, resulting in this shift, and that is called as bathochromic shift or the red shift. It can be done by, as I said, oxochrome, or there could be other reason for that. For example, the change of the solvent. So we have already taken the example of the oxochrome group like hydroxyl or OCH3 group or NH2 group in case of the aniline you just studied, that results in the bathochromic shift or the red shift. Uh, in the alkaline medium, as I said, the solvent play a role. In alkaline medium, p-nitrophenol shows a red shift because of the fact that negatively charged oxygen delocalizes because of alkaline medium more effectively than unshared pair of electron. And you can see here that this p-nitrophenol has a maximum absorption at 255 nanometer. But when we change the environment, we change the solvent to alkaline medium. As a result of this, you can see there is a change in the structure. And as a result of the change in the structure of this, oxygen, which is negatively charged now, the maximum absorption jumped from 255 to 265, and that's what the increase in the absorption wavelength, and that is called as bathochromic shift. So you got an example that the solvent can have this effect to do the bathochromic shift, and the addition of the oxochrome will also result in the same. The second type is called as the hypsochromic shift, which is the blue shift. Now, what is hypsochromic itself? When absorption maxima of a compound shifts to a shorter wavelength, it's just opposite of the red shift. So in this case, there is a shift of the wavelength on the shorter side, and that's why it is called as hypsochromic shift or the blue shift. Now, this effect is due to the presence of a group, or that because of the removal of a conjugation or by the change of the solvent. So there are different aspects of this hypsochromic shift. It could be because of the presence of a group, because of the removal of a conjugation, or simply by the change of the solvent. So here are some examples. One example is of the aniline. Now, you studied that aniline is basically a benzene with the amine group there, and the absorption maximum was 280 nanometer. When we change the medium, the solvent actually, and that's an acidic medium, as a result of this, this is being changed to NH3 plus Cl negative. And you can see that the absorption maximum is decreased from 280 to 265 nanometers. So that's what the decline or towards the shorter wavelength, and that's called the hypsochromic or the blue shift. The third one is the hyperchromic effect. Now, what is hyperchromic effect? When absorption intensity of a compound is increased, it is known as hyperchromic shift. Now, we are not talking about the shift of the absorption wavelength. Here we are talking about the change of the absorption intensity itself. This is a different aspect. That's why it's called hyperchromic effect. So let's go to see some of the examples that how this hyperchromic shift takes place. For example, if we have an oxochrome and we introduce that to a compound, the intensity of the absorption increase. Examples are there. The example of the compound pyridine. Pyridine has an absorption maximum at 257, and as a result of the incorporation of a methyl group, it becomes 2-methylpyridine, and now the absorption maximum is at 260 nanometer. Now, this is a very slight change of the absorption maximum, but actually this is because of the fact that introduction of the oxochrome results in increase of the absorption intensity itself by the compound. The last one is the hypochromic effect. I'm sure that you can have understanding of this. This is the reverse of the previous one, which was hyperchromic. You might have this consideration that in this we have a change in the wavelength as well. So this, we are not talking about the red or the blue shift. What we are talking about, as I said, we're not talking about the change of the maximum absorption wavelength. We are talking about the change of absorption intensity itself by the compound, and that's what the concept of hyperchromic and hypochromic effect is. So in hypochromic effect, when the absorption intensity of a compound is decreased, that is being known as hypochromic shift. And the examples are here: naphthalene and 2-methyl naphthalene. There's the incorporation of methyl group, and you can see that we have a sharp decrease of absorption intensity from 19,000 to 10,250. So that's we call as hypochromic shift or hypochromic effect. So we got four things here that we have discussed: bathochromic, hypsochromic, and then hyperchromic and hypochromic itself. Now, this is a graph which shows the relationship between absorbance versus the wavelength. And this is, I would say, the summary of what we studied right now. With the increase of the absorption and the increase of the wavelength, we can see that more light is going to absorb till it reaches this point, and then it starts to decrease. So at this point, you can see there's the maximum absorbance, and this is the particular wavelength which shows the maximum absorbance. Now from their point onward, if there is an increase in the absorption, like for example because of the incorporation of a compound, this graph will be slightly like going this way and going up top like this, and then going down. That's called hyperchromy. If the incorporation of a functional group or any substitution would result in this graph with a lower intensity going like this and coming from their point, this is called hypochrome. Similarly, this particular wavelength is the wavelength at which we have the maximum absorption. And if this maximum absorption increases because of any compound, is called redshift. And if this is decreased because of the incorporation of any functional group, that's called blue shift. So this picture is itself the summary of all these four shifts and effects that we have discussed. Now, finally, coming back to the conclusion of the topic, to end up the topic with some of the applications of UV visible spectroscopy. There are many applications in the biochemical sciences. Few of them are being listed here. The first application is about qualitative and quantitative analysis. We know that there are two primary bases of doing the analysis, either on the basis of the quality or on the basis of the quantity. So the UV visible spectroscopy can be applied for both qualitative and quantitative analysis. And I'm sure that you guys remember the spectroscopy that you guys have done in your uh, uh, BCH 411 and BCH 412, where mainly you use the visible spectroscopy for the quantitative analysis. Now, for this, it is used for either characterizing the aromatic compounds and their conjugated olefins and their double bonds, or it can be used to find out the concentration. As I just mentioned, in 411 and 412 you have done the molar concentration, you have determined glucose, concentration of proteins, and cholesterol, and so on and so forth. So this is both qualitative and quantitative use of the technique. Then this technique can also be used for the detection of impurities, because impurities will indirectly affect the absorption capacity of a compound. And when the presence of an impurity results in change of the absorption, that can be determined by this technique to see that whether this compound is pure or impure. So it is one of the important methods to detect impurities, mainly in the organic solvents. This technique of UV visual spectroscopy can also be used for the detection of isomers, because different isomers have got different configurations, and because of different configurations they would have different absorption capacities at specific wavelengths. So we can differentiate between two different isomers based on the knowledge that what is their maximum absorption wavelength, and we can determine about that. Then determination of the molecular weight by using the Beer law that we have already studied. So this is all about UV visible spectroscopy. With this, we finish the lecture of UV visible spectroscopy. And inshallah, in our next class, we're going to study about our next technique, which is infrared spectroscopy. So I'm going to upload this lecture, and in the meanwhile, if you have got any question, you can ask your question in person or in the group, and I'm more than happy to give you the answers. Thank you very much, and inshallah, see you for the next class. For the office.